Hello children, how are you fine? Hope you are enjoying my class and exploiting maximum benefit out of my video lesson. So today we are progressing towards the third chapter, human reproduction, and I will be dealing with you today the process of spermatogenesis, that is the process of formation of sperm in case of human male. But before we go in detail about the process of spermatogenesis, let us take a section TS through testis, where the so many seminiferous tubule with the testicular lobe you will see, and you will see the location of different cells where from the process of spermatogenesis starts. So let us draw a cross section or TS of testis. Suppose this is, we have cut a cross section, transverse section TS of testis, where you are getting so many fine tubular structure in cross section which almost look like this. Say suppose some of the tubular fine structures got a half cross, some are appearing entirely through or partially completed. These are actually the seminiferous tubule, that means the section gone through the seminiferous tubule, and inside the seminiferous tubule one I will draw. You have the basement membrane layer, This is the basement membrane layer I am drawing. After that I level. Then we have different size cells. These are the spermatogonial cell. Otherwise we can call spermatogonia. Then we have certain other cells. This is what we call sertoli cell. And we have immature sperm, that is spermatid, and we have spermatozoa, or called mature sperm. So the same structure you will get everywhere within this cross section, passing through the seminiferous tubule, the spermatogonial cell, the sertoli cell, spermatid, and spermatozoa. So these structures are the seminiferous tubule. Within the seminiferous tubule you have the basement membrane, membrane, we have these cells called spermatogonial cell or spermatogonia. Spermatogonia or spermatogonial cell, we have these cells called sertoli cells, and the spermatozoa or called mature spermatozoa, and these are the spermatid or immature spermatid. And in between the seminiferous tubule, in between, this is 1, 2, 3, 4, 5, 6. Suppose 6 seminiferous tubule through which the section have gone. In between the seminiferous tubule, these spaces are filled with clusters of cells, either remaining cluster or in separate way. Individually, these clusters of cells, these are called leydig cell. Leydig cell, otherwise called interstitial, otherwise leydig cell, otherwise called interstitial cell of leydig. Interstitial cell of leydig. So when we have gone through a TS, transverse section through testis, we found the seminiferous tubule. Within the seminiferous tubule, we have seen the basement membrane, we have seen the spermatogonial cell, we have seen the sertoli cell, the immature sperm, otherwise called spermatid, and mature sperm, spermatozoa. In between the seminiferous tubule, the spaces are filled with clusters of cells called leydig cell. These are the leydig cell, otherwise called interstitial cell of leydig. Once we got this concept very clear, then we can pass on to the spermatogenesis. But before I dealt with spermatogenesis, few things more we shall have to know. What are the things? So if I rub this part, see the interstitial cell of leydig or leydig cell. Leydig cell is very important indirectly for the process of spermatogenesis. Why? Because it is the leydig cell which produces male androgen called testosterone. This production of testosterone influence the process of spermatogenesis. So it is the testosterone hormone produced from leydig cell. When the testosterone hormone, otherwise called male androgen, starts producing, the process of spermatogenesis onset, and the production of testosterone hormone by leydig cell, again guided by another hormone called leydig, luteinizing hormone. Two hormones, FSH, follicle stimulating hormone, FSH, what is it called? Follicle stimulating hormone, and LH, luteinizing hormone. Especially LH, it is having a direct impact on the leydig cell for the production of testosterone. Where from this hormone secretes? This is secreted by anterior pituitary gland. Anterior pituitary gland, that is otherwise called the master gland. Anterior pituitary gland. This FSH and LH secreted by anterior pituitary gland, and the hormone production, the FSH and LH from the anterior pituitary gland, again guided by the gonadotropin that is produced from the hypothalamus. So this gonadotropin from hypothalamus stimulates the production of FSH and LH. LH in turn secreted from the, both are secreted from the anterior pituitary gland, stimulates the production of testosterone from the leydig cell, and it is the testosterone hormone influences the process of spermatogenesis. Now we shall switch on to spermatogenesis. By that time, the sectional view of testis, the different cells present in the seminiferous tubule, and in between the seminiferous tubule, the leydig cell and function of leydig cell, I hope it is very, very clear to you. So with this concept, now we shall switch over to the next process, that is process of spermatogenesis, that is a part of gametogenesis. In case of male, the gamete sperm production, that is what we call as spermatogenesis, it is a part of gametogenesis. So let us talk about the spermatogenesis. So let me rub this part. And this level also. So spermatogenesis. Spermatogenesis. What is spermatogenesis? It is the process of formation of spermatozoa, otherwise called sperm. It is the process of formation of spermatozoa from the spermatogonial cell. Spermatogonial cell. Where does this spermatogonial cell present? It present within the seminiferous tubule. So this process takes place within the seminiferous tubule. The process of formation of spermatozoa from the spermatogonial cell of where? Seminiferous tubule or seminiferous seminiferous tubule is called spermatogenesis. So it is the process of formation of spermatozoa from the spermatogonial cell of seminiferous tubule. Is that clear to you? Now let me draw a half section, that is cross section through one such seminiferous tubule, and stepwise explain the different events takes place for the formation of spermatozoa. So let me wrap this part. If I take an enlarged view of cross section through one seminiferous tubule, if it is looking like this, the basement cells inner to the lining of the seminiferous tubule, the basement cells on which lies the spermatogonial stem cell. Spermatogonial stem cell. This thing is a little new concept, not given detail in your NCERT textbook, but you add here that what is stem cell? Stem cell is such a cell which is potentially active for giving rise to any kind of cell. So the spermatogonial stem cell can give rise to a functional spermatogonial cell by differentiation process. This spermatogonial stem cell give rise to two type of cell, A cell and B cell. A cell again give rise to spermatogonial stem cell, whereas B cell modifies to form spermatogonia. So suppose this is now produced from the spermatogonial stem cell B type. So this will be called as, so what do you call this? We'll call this as, and so many will be produced like this from this cell. This will be called as spermatogonia, otherwise called spermatogonial cell. Otherwise, commonly can be called as sperm mother cell, spermatogonia. So, what will be the ploidy of this cell? Diploid 2n, that is having 46 number of chromosome, 46 chromosomes. That is what you call diploid set, two sets of chromosomes, that is 46 in number. So these spermatogonial cells are by nature diploid. They will produce on sequential event to the sperm. That is why otherwise we can call this as sperm mother cell. Is that clear? Now, another type of cell also present within the seminiferous tubule, another type of cell, these cells are called sertoli cell. Sertoli cell. So they are called sertoli cell. Sertoli cell. Now, what happened? These spermatogonial cell or sperm mother cell, if I rub this side, so this spermatogonial cell which is by nature diploid, that is having 46 chromosome, they will undergo mitotic cell division. What is that cell division? Mitosis. And they will form primary spermatocyte. Primary spermatocyte. This is a mitotic division. So mitotic division is equal division. That means chromosome number will remain. Yes, chromosome number will remain same. So there will be no change in chromosome number 2n. That is 46. Now it is this primary spermatocyte. They will undergo meiotic division. Meiotic 1 and 2. As you know, meiosis is having two divisions, meiosis 1 and meiosis 2. It will undergo first meiosis 1. By the process of meiosis 1, primary spermatocyte, each primary spermatocyte will divide to form two cells. What are the two cells? Secondary spermatocyte. What will be the ploidy? Meiosis. This is meiosis 1. Meiosis 1. First meiotic division. The product will be resultant product, that is secondary spermatocyte will be having haploid set of chromosomes n number. That is 23. Then the secondary spermatocyte will now undergo second meiotic division, and when the second meiotic division will complete, that is meiosis 2. Meiosis 2. This is meiosis 1, this is meiosis 1, this is meiosis 2. By the end of second meiotic division, each secondary spermatocyte will divide to form two cells. Each are called spermatid. Spermatid. So what will be the ploidy of spermatid? Again N. Because of second meiotic division is just like the mitosis. The second meiotic division, the number of chromosome again retains same. That is 23. Ploidy N, chromosome number 23. What is this spermatid? Spermatid actually immature sperm. It is otherwise called immature sperm. This spermatid will now undergo a process of change that I will discuss in detail, and by that change it will mature to form spermatozoa. What is it called? Spermatozoa. and the process by means of which immature sperm or spermatid gets converted into spermatozoa. That process is called spermiogenesis. Spermiogenesis. So if I use other chalk color, this process is called spermiogenesis. This process is called spermiogenesis. So what is spermiogenesis? It is a process by means of which the spermatid or immature sperm getting converted into a mature sperm, otherwise called spermatozoa. Spermatozoa is nothing but a mature sperm. So we can write here mature sperm. Now the question is, how the immature sperm getting converted into mature sperm? By that time, by the end of second meiotic division, when the spermatid show form, they do not have any functional tail. How the tail is formed? So the sperm head, by that time, if I draw the diagram here, by that time the sperm head made up of nucleus, the sperm nucleus become condensed, packaging of chromatin material takes place, and the Golgi body gets transformed into a cap-like structure over the sperm head, called acrosomal vesicle. Acrosomal vesicle. So if I rub this part, or if I rub right here, this is the acrosomal vesicle or acrosome cap. Acrosome cap over the sperm head. The nucleus becomes very condensed, the chromatin material gets packaging, the other extra cytoplasmic parts and unnecessary cell organelles are withdrawn, leaving behind the major part, that is the sperm nucleus present within the sperm head, and microtubule develops from one of the centriole. These microtubules are forming the cytoskeleton part, which immediately next to its sperm head having a little notch-like part, and then the microtubule proceed further. The anterior part of the microtubule, gradually the anterior, let me little modify. The anterior part of the microtubule is quite wide, and wide is wide because of the arrangement of mitochondria. Mitochondria will arrange over it in the form of bands. These are the mitochondria. These are the mitochondria. And now gradually it becomes narrow. To form a So this is the middle piece, this is the head of sperm, this is the head part, head, having a coverage of acrosomal cap, having a centrally placed nucleus, a middle piece, and a tail. So these transformation taking place during spermiogenesis. So spermiogenesis is the transformation of immature sperm, otherwise called spermatid, into a mature sperm, otherwise called spermatozoa. Now it is called spermatozoa, having a sperm head, middle piece, tail, middle piece filled with mitochondria, which is the active side, giving energy. Now it is a fully mature sperm. This is the structure of sperm, human sperm. Now, once the immature sperm, by the end of meiosis II, immature sperm or spermatid so form, they remain picked within the sertoli cell. These are the sertoli cell. Within the sertoli cell. Say if I draw it like this. Within the sertoli cell. Within the sertoli cell. Now, I told you two hormones. One is FSH, and the other is LH. It is the luteinizing hormone which helps for the production of testosterone. What is the role of FSH hormone? It will promote the sertoli cell to produce certain factors which otherwise help for this sperm maturation. And by the end of spermiogenesis, the immature sperm getting a acrosomal cap, then middle piece filled with mitochondria and the functional tail. Now it is becoming spermatozoa, otherwise called functional sperm. These are the spermatozoa. So spermatozoa or mature sperm. Once the spermatozoa so form, they come within the lumen. If I make a circle of this seminiferous tubule, then they will come towards the lumen. If I draw another, so they will come. This is the spermatogonial cell. These are the sertoli cell. These are the sertoli cell. These are the basement membrane. Why I am drawing again? So as to give you the clear concept. When the sperm, mature sperm produce, where do they come? Where do they come? This is the basement membrane. This is the basement membrane. Now, this is the spermatogonial cell. These are the sertoli cell. sertoli cell. Once the sperm become mature, get a complete new fully matured tail with sperm head covered by a chromosomal cap. It is now migrating towards the lumen of the seminiferous tubule. This is the lumen of the seminiferous tubule. Now when they are coming to the lumen of the seminiferous tubule, now they are ready to expel from the seminiferous tubule. But by that time they are not motile, although they are having functional tail, they are not motile. From the seminiferous tubule, they will enter to the epididymis. As I told you, very coiled structure at the anterior end of the testis. They will enter to the epididymis, and here they gain their motility. So in that way, the whole process of spermatogenesis is completed. So the main aim of spermatogenesis is the formation of a mature sperm. So if I make a synopsis of the whole thing we have discussed today, we have made a cross section through testis. If we make a cross section through testis, we will get so many seminiferous tubule. Seminiferous tubule are the site for the production of sperm. Within the inner layer of seminiferous tubule, there is a basement membrane, and next to the basement membrane we have spermatogonial stem cell. These spermatogonial stem cell gives two types of cell, type A cell, type B cell. Type A cell again give rise to spermatogonial stem cell, type B cell give rise to spermatogonial cell, otherwise called sperm mother cell or spermatogonia, and sertoli cell. It is this spermatogonia or sperm mother cell undergo a sequential events of mitosis followed by meiosis 1 followed by meiosis 2 to form immature sperm. Spermatogonia, which is having diploid set of chromosome 46 number, undergo first mitotic division, thereby producing primary spermatocyte. Primary spermatocyte is having the, what is the ploidy? Diploid, 2n number of chromosomes, that is 46. As the spermatogonia is also having the 46 chromosome. Then the primary spermatocyte will undergo meiosis 1. By the end of meiosis 1, they will produce secondary spermatocyte. From one primary spermatocyte, two secondary spermatocyte will produce, and it's a reductional division having number of chromosome not diploid, two sets, single basic set of chromosome 23 in number. Then the secondary spermatocyte undergone meiotic 2 or second meiotic division or meiosis 2 to form spermatid. Spermatid is the immature sperm, same chromosome number 23. Immature sperm undergo a process of spermatogenesis to form mature sperm, otherwise called spermatozoa. Once the spermatozoa is formed, they come within the lumen of the seminiferous tubule, and from the lumen now they are ready to enter to the epididymis. This process of release of sperm into the lumen after getting maturation is called spermiation. So the whole process of spermatogenesis I have discussed. I have discussed the shape of a mature sperm, what are the parts, centrally placed nucleus, almost oval in shape, covering almost entire area of cytoplasm, covered by a chromosomal cap. This is very helpful for penetrating through the different layers of ovum boundary, having a middle piece filled with mitochondria, supplies the required energy for its motility and the tail, the hind part. So I hope you have got a very clear concept of testis, its cross section, the different parts, where the process of spermatogenesis takes place. What are the steps of spermatogenesis? What is spermiogenesis? What is spermiation? And what are the event takes place during sperm maturation or spermiogenesis? So, is it very clear to you? I hope you got a very clear concept about spermatogenesis. With this, thank you.